Welcome to Chromo Chat. Today we're going to be talking about Huntington's disease. This is a rare neurodegenerative disease that causes nerve cells in the brain to die and lose their function. Other neurodegenerative diseases you have probably heard of are Alzheimer's and Parkinson's. So what is Huntington's disease? To be able to understand, we must first take a look at some basics of genetics. This will allow you to understand what causes this hereditary disease. Imagine the entire human genome as a massive library that contains all the information needed to build and maintain an organism such as a human. Every single person has a genome that is unique to them. Deoxyribonucleic acid, also known as DNA, is the language in which the books in the library are written. This language is composed of sequences of 4 different letters: A for adenine, T for thymine, C for cytosine, and G for guanine. These letters are arranged in specific sequences to form words, sentences, and paragraphs. Just like how letters in a book form words, sentences, and paragraphs. Codons can be seen as words in the language of DNA, and each codon is made up of a sequence of 3 letters from the DNA alphabet: U, C, A, G. For example, the codon CAG represents the amino acid glutamine. Amino acids are the building blocks of proteins, as they contribute to the structure, function, and regulation of the protein it codes for. A gene is a segment of DNA that carries the code for a particular protein or a specific function. Genes are found in chromosomes, which are structures made up of DNA. Each chromosome contains many genes. In humans, the DNA in each cell is organized into 46 chromosomes, or 23 pairs. When these proteins fold improperly or misfold, it leads to the formation of a dysfunctional protein. There are various factors that can cause a protein dysfunction, one of them being genetic mutations, which is the cause of Huntington's disease. The HTT gene is found on chromosome 4, and it provides instructions for making the protein Huntingtin. This protein can be found in many of the body's tissues, with the highest levels of activity in the brain. As stated before, the codon CAG codes for the amino acid glutamine. The HTT gene contains a tail, where CAG sequences are repeated 10 to 35 times. If the HTT gene is mutated, more CAG trinucleotide repeats will form on the tail. If the gene has too many CAG repeats, it becomes abnormally long. This makes the Huntingtin protein misfold, and since the structure of a protein is imperative to its function, the function is affected. The more CAG repeats there are, the more affected a person is. Less than 26 or up to 35 repeats have no effect on a person, whereas 36 to 40 CAG repeats show reduced penetrance. 40 or more repeats shows complete penetrance, indicating the person has Huntington's disease. The mutated protein aggregates within the neuronal cells of the caudate and putamen regions of the basal ganglia. The caudate region is responsible for the planning and execution of movement, learning, memory, reward, motivation, and emotion. The putamen region is responsible for motor control, speech articulation, language functions, and overall cognitive functioning. When proteins aggregate in this area, neuronal cell death is caused. Thank you for watching. Don't forget to subscribe and join us for another Chromo Chat.